Gabrielle, you're admitting everybody. Good morning, everyone. We'll get started in just one moment, giving people time to get signed on, and then we'll get started. Okay, well, we'll go ahead and get started. Hello, everyone, and welcome to IAC for Children's educational webinar on pediatric cell and gene therapy. We're delighted you're here, especially if you're joining us for the first time. My name is Angie Price. I'm the Director of Education and Marketing Communication, and I'll be your moderator for today's webinar. I look forward to spending the next 60 minutes with you, exploring the science behind pediatric cell and gene therapy, along with the ongoing ethical challenges. Before we get started, I'd like to cover a few housekeeping items. Today's webinar is being recorded. We will be providing the recording and a copy of the slides within the next week. We encourage you to use the chat feature to ask questions, and we will do our best to answer those questions at the end of the speaker's presentation during our Q&A. For those of you that don't know about IACT, we are an organization with a mission of urgency. We work with patients, sites, and pharmaceutical companies to expedite pediatric research. Our support extends to over 70 trial-ready sites where we provide clinical trial opportunities, mentoring, quality improvement initiatives, regulatory support, and ongoing educational webinars like this one. We also collaborate with pharmaceutical companies on pediatric clinical trial designs, feasibility projects, site identification, and initiatives to advance pediatric research. Our vision is to reduce the time it takes for pediatric drug development from 9 to 5 years, because I think we all can understand that children do not have the time to wait. So here are our objectives for today. I think that we can all agree that cell and gene therapy is pretty complex, but as researchers, it's crucial that we have a basic understanding of the science behind it, the applications, as well as the ongoing ethical issues. This knowledge can mitigate misunderstandings or misconceptions about cell and gene therapy, especially with families that we need to educate so they can be prepared to make informed decisions about potentially having their child participate in cell and gene therapy. And now it's my pleasure to tell you a little bit about our speakers. First, we have Merritt Siverston. First, I would like to introduce her. She is the mother of a young boy with Duchenne's muscular dystrophy and a DMD parent advocate. Merritt is a health law attorney by trade with vast healthcare knowledge and expertise. She is passionate about advocating for health professional clients and defending their rights. Merritt applies these talents as a parent to advocate for Duchenne. We are truly grateful to have Merritt here today to share her son's journey with cell and gene therapy. Next, we have Dan Eisman, the Executive Director of Biosafety Services at Abera, where he oversees the Institutional Biosafety Committee and provides biosafety consulting services to researchers on regulatory requirements, ensuring they conduct research safely and with minimal regulatory delays. He holds a PhD in molecular biology and immunology. Dan has a passion for education and has taught microbiology at both the undergraduate and graduate levels. It is an honor to have Dan with us today. I wish he'd been my teacher on microbiology. Lastly, we're excited to welcome Carolyn Chapman. Carolyn leads the Multi-Regional Clinical Trial Center at Brigham in the Women's Hospital at Harvard. Carolyn is also a lead investigator in the Division of Global Health Equity. Carolyn holds a PhD in genetics and a master's in bioethics. Her extensive expertise in research ethics and genetics promises to bring a wealth of knowledge to today's webinar. With that, I will pause. Has Merritt been able to join? Okay. So we're going to shift out of order. I'm sure our busy Duchenne's parent is probably handling a crisis. So we're going to move into having Dan take over. I'm going to briefly move to his presentation.
Great, thank you. Happy to jump in. So
Thank
welcome
you.
everyone. I am Dan Eisenman, Executive Director of Biosafety Services at Advara. I'll introduce the types of gene therapy approaches utilized today that allow for exciting medical advancements. I'll also introduce some of the associated challenges and risks. So one of the take-home messages of this presentation is the diverse types of technologies and approaches utilized in gene therapy. Each tool in the toolbox has different potential uses and potential for risks. As such, it is important to consider the unique uses and risks rather than using a one-size-fits-all approach when evaluating gene therapy. So the most common approaches we're currently seeing include gene-modified cellular therapies, genetic vaccines, gene transfer, oncolytics, which is predominantly about using viruses to kill cancer cells, gene editing, and gene-modified bacteria or bacteriophages. This field is quickly growing. This is a figure from a paper we published in Applied Biosafety, which was selected for the cover. It shows the number of investigational new drugs issued by the FDA specifically for gene therapy. This figure illustrates how gene therapy is growing quickly. However, this figure also tells the story of some of the major challenges that needed to be overcome. The gene therapy trials grew at a steady rate in the 90s, but hit some major challenges around the turn of the century with high-profile serious adverse events. Jesse Gelsinger died after receiving the highest dose of an adenovirus vector in a phase 1 dose escalation trial for a rare metabolic disease. Just a few years later, there were a number of pediatric trials that had to be halted as gene delivery using retroviruses into bone marrow led to leukemias. In a few slides, I'll discuss why gene therapy frequently involves the use of genetically modified viruses. Now, over the next decade, the number of trials plateaued as the field focused on studying the risks associated with these technologies and improving their safety profiles. Now, around 2015, the conditions were right for a resurgence. The first FDA-approved approval for gene therapy was issued in 2015 for a gene-modified herpes virus for use in melanoma. 2017 saw 2 approvals for gene-modified cellular therapies for use in B-cell leukemias. And finally, by 2018, we saw that gene therapy had come of age with an increasing number of new drugs and a steady stream of FDA approvals. Now, not long ago, the idea of having an FDA-approved gene therapy felt like impossible science fiction. The field has overcome multiple challenges, and we now see several approvals per year. Initially, oncology led the way. There was a surge of infectious disease research and approvals that coincided with the COVID-19 pandemic. And we're currently seeing a surge in rare disease research, which will likely outpace approvals for all other disease indications. Before we get into applications of the technology, let's provide a quick intro to how this technology works. So let's say the red DNA codes for a potentially therapeutic protein. That DNA needs to be introduced into a delivery vehicle to help get it into the cells to make that therapeutic protein. The simplest form is a plasmid, or simply put, a circle of DNA. We can splice our therapeutic gene of interest into the plasmid to make a very simple gene therapy product. Now, this approach is very low risk, but naked DNA by itself is not easily taken up by cells, especially human cells, which can affect efficacy. So scientists look to nature to find better gene delivery vehicles. If we look at the viral life cycle, infection involves delivery of the viral gene to host cells, in order to take advantage of the host's biosynthetic machinery and allow viral replication. So if we remove the genes involved in viral replication and disease, what we're left with is essentially a virus that can only bind to the target cell and deliver genetic material. It functions very much like a genetic syringe. And this is what we refer to as a viral vector. Viral vectors allow us to easily introduce genetic material into target cells during infection. While the viral vectors are typically unable to replicate, they are still infectious and must be handled accordingly. Now, this figure shows the various animal virus families. Viruses encompass a great deal of diversity, which gives potential for a large number of potential uses and risks. There are also diverse possibilities for toxicities. It is important to have a solid background in virology for review of virus-based studies. Now, let's discuss some various pediatric diseases and see how gene therapy could be used, but also what challenges can arise. So with Duchenne muscular dystrophy, cystic fibrosis, and sickle cell disease, these are all monogenic diseases, or diseases that are caused by mutations to single genes, respectively. Our current technology is quite good at delivering single therapeutic genes. Therefore, these types of rare diseases are good targets for this technology. However, each disease also poses challenges. Duchenne's would require delivery of enough of the gene therapy product into the major muscle groups to correct function. The symptoms of cystic fibrosis are most severe in the lungs, which means we'd need to deliver an aerosolized gene therapy product into the lungs. The disease process also produces thick mucus in the lungs, which can interfere with access to the cells of the lungs, which the drug would need to have access to those cells that are protected by the mucus. Cancers frequently stop expressing proteins that allow the immune system to attack them, thereby evading immunological surveillance. Chimeric antigen receptor T cells, or CAR T cells, provide a means of attacking the cancer cells directly despite their aims to evade immunological surveillance. And then lastly, in sickle cell disease, a mutation causes red blood cells to form a sickle shape, which can lead to painful and dangerous blockages. Since all the cells in the blood originate from bone marrow stem cells, a gene therapy for sickle cell would require delivery into bone marrow stem cells, which would then need to grow in the body and produce the red blood cells needed to correct the disease. There we go. Sorry, the slide was slow in advancing. So this slide provides a handy reference for the most commonly utilized gene delivery vectors. There are two properties that I'd like our audience to consider as we discuss these vectors over the rest of my presentation. First is, does the vector integrate into cellular DNA? In other words, does it insert itself into the DNA of the subject cells? The second consideration is, does the vector trigger an immune response? And I'll elaborate on why these are important considerations shortly. Retroviral vectors are quite good at inserting themselves into cellular DNA. Genomic integration has some beneficial properties. The DNA encoding the potentially therapeutic protein is permanently inserted into cellular DNA, which means the transgene is passed on during cellular proliferation, as is needed for cells that require proliferation, such as chimeric antigen receptor T cells, as well as stem cells. Now, let's talk about some of the risks associated with gene therapy trials. So sticking with genomic integration, It's permanent. There's no undo button. The vector integrates randomly into cellular DNA. Now consider 85% of the human genome does not code for anything, so integration there should be safe. However, insertion into the 15% of the genome that contains gene coding sequences can cause mutation or even increase cancer risk, which is why use of integrating vectors requires long-term follow-up. The need for long-term follow-up for integrating vectors is highlighted by recent news about the FDA-approved treatment for cerebral adrenoleukodystrophy, which is a fatal neurologic disease. So out of 67 patients that were dosed at that time, 7 developed blood cancers, 1 died, and more could develop similar SAEs over time, hence the need for long-term follow-up. A type of gene-modified cellular therapy called chimeric antigen receptor T cells, or CAR T cells, have revolutionized treatment of B-cell leukemias and lymphomas that previously were considered resistant or refractory. So CAR T cells are manufactured using integrating vectors, so the therapeutic transgene stays in the cells as they proliferate. However, the FDA had to investigate reports of secondary leukemias following treatment. The FDA found that the CAR T cell transgene in some of the secondary leukemias. In those cases, the CAR T cells were becoming cancerous. Other secondary cancers may have also been due to the preconditioning chemotherapy that is required as part of the treatment. Now, the next few slides are intended to show that even non-integrating vectors can bear some risk. So, for example, adeno-associated viral vectors, or AAV, are frequently considered one of the safest vectors and serve as the workhorse for gene therapy into non-dividing cells. The virus does not integrate. However, for gene delivery, oftentimes the virus needs to be concentrated into ultra-high doses to achieve efficacy. In subjects that are frail enough, that have certain comorbidities, or far enough along in the disease process, even AAVs can cause serious adverse events, including death. Another virus, adenovirus, is another type of non-integrating virus. It is highly immunogenic. If the vector triggers an immune response, it can make repeat dosing challenging. The subject might have prior immunity to the vector, which can decrease the efficacy of the product. So consider that what if the virus is in the environment and the subject previously came in contact with it before participating in the trial, they could have neutralizing antibodies in their blood, which would limit the efficacy of the product. Earlier I mentioned the death of Jesse Gelsinger. So Jesse Gelsinger received the highest dose of an adenoviral vector in a phase 1 study for a gene therapy trial for a rare metabolic disease. He died from an acute immune response to the adenovirus vector that was delivered into his liver. Now let's return to the various pediatric diseases I mentioned earlier. All of these diseases currently have gene therapy approaches in use, either in clinical trials or as FDA-approved products. So I showed earlier the case study where a subject with Duchenne's passed away. Another concern is producing enough of the gene therapy product to deliver the therapeutic gene into the major muscle groups to correct the function. This is not a trivial task. It's quite challenging to manufacture that much virus. And then factor in the various limitations that come with manufacturing and providing it to the subject in a timely manner. The virus is moderately immunogenic and present in the environment, so pre-existing neutralizing antibodies can decrease efficacy. And the immunogenicity of the virus also complicates repeat dosing. Different approaches are being tried for cystic fibrosis. Delivery into the lungs is complicated. Also, in terms of site suitability, A research site working on these trials would require the right facilities, ventilation, and respiratory protection to protect the research staff administering the aerosolized investigational product. B-cell leukemias were considered resistant or refractory to treatment just a decade ago, are now often treatable with chimeric antigen receptor T cells. These CAR T cells frequently exhibit high overall response rates. However, they are manufactured with integrating vectors, so long-term follow-up is required to assess long-term cancer risk. And lastly, with sickle cell, there are currently 2 approved therapies for sickle cell disease. One involves an integrating vector, and the other involves gene editing. So, of course, both require long-term follow-up.
Thank you, Dan, so much for giving us that overview. I think that you have a wonderful way of taking a very complex cell and gene therapy and kind of making it easier for the lay person to understand. I think it's important to understand the applications. A lot of people get into their own, what they're researching, and kind of like expand that and see, where it is touching and really how far we have come. So I'm pleased that Merritt has joined us. We're excited to have her tell her story. We'll have her go after Carolyn talks about the ethics of cell and gene therapy. So I would like to turn it over to Carolyn. This is a crucial topic about ethics. It requires everyone to have knowledge of this so we potentially avoid danger to our patients and lastly, jeopardizing our community and how they trust in the research that we're doing. Thank you, Carolyn. Glad that you're here today.
Thank you. Thank you so much, Angie. I'm really, it's an honor for me to be here. And thank you, everyone, for joining today. As Angie said, I lead the Cell and Gene Therapies Project at the MRCT Center, or the Multi-Regional Clinical Trials Center of Brigham and Women's Hospital in Harvard. And we are basically a policy center that focuses on addressing challenges in the conduct, ethics, oversight, and regulatory environment for global clinical trials. And this is just our standard disclaimer slide. I just want to mention that what I expressed today, my opinions are my own, and also just point out that the MRCT Center is supported by voluntary contributions and grants from foundations, corporations, international organizations, as well as government entities. And so, at the MRCT Center, we're also focused on issues and challenges in the context of clinical trials. And so, as Angie said, I'm going to focus on ethical issues that arise in the specific context of cell and gene therapies. But before I do that, I think it's important maybe to start out by touching briefly on the ethical framework that undergirds all types of research with humans. And for some of you, this might be, old hat and you already know, but I just want to make sure that everybody's on the same page. So in the 1970s, in the wake of the Tuskegee research scandal, Congress established the National Commission for the Protection of Human Subjects. And tasked that committee with identifying ethical principles and guidelines to guide biomedical and behavioral research involving human subjects. The output of that committee, famously, was the Belmont Report, which came up with 3 overarching ethical principles: respect for persons, often thought of as autonomy, beneficence, and justice, which together drive requirements for the ethical conduct of research. Respect of persons necessitates a robust informed consent process, as well as protection for people without capacity to make decisions for themselves, such as potentially pediatric patients, although they obviously have some capacity as they get older to assent to procedures for sure. Beneficence, or simply put, helping and doing good for others, requires that risk to participants are minimized and justified by potential benefits to the research participants and to society. And then justice requires fair selection of research participants and equitable distribution of risks and benefits of the research. So these ethical principles and requirements became foundational and were basically promulgated into the U.S. federal regulations as well as regulations around the world. So, okay. So although all people involved with conducting research with humans must adhere to these ethical principles and regulations, including researchers and clinical staff, clinical research staff, and a whole variety of people in this ecosystem, one key checkpoint is the involvement of institutional review boards or ethics committees, which review research protocols to protect the rights and welfare of research participants. IRBs review the risks and benefits of protocols, the informed consent process, qualifications of investigators, and ensure that participants are recruited fairly. And, appropriate informed consent involves providing information that a reasonable volunteer would want to know in making their decision, confirming understanding of this information, and making sure participation is voluntary and not coerced in any way. So we know that there are ethical issues involved with all types of research involving humans, and we do have mechanisms in place to support the ethical conduct of research with humans. So what makes cell and gene therapy clinical research so different or special? So I think that there's a few reasons. And, Dan's introduction provided sort of a precursor to thinking about the ethical issues, because it really depends on one of the big reasons that there's heightened ethical challenges in this context is the actual characteristics of cell and gene therapies, their scientific mechanism of action and delivery, which Dan mentioned. But also he mentioned, that they can have complex manufacturing and supply chains. And because cell and gene therapies are a relatively new therapeutic class, there are also uncertainties in addition to these complexities. And then another factor is the disease patient context, which Dan also started to get into. So because of that, in totality, all of these factors come together and make navigating the ethical requirements for the design and conduct of cell and gene therapy research, and potentially even the delivery of cell and gene therapies in clinical care, a bit more challenging. So I wanted to spend a little bit more time digging into why this is the case. So first, let's consider in a little bit more detail, and again, this might be reviewing what Dan said, but putting like an ethics spin on it, these unique characteristics of gene therapies. So one issue is that gene therapies, particularly those which modify, replace, or add DNA, are often intended to make permanent or long-lasting changes to human cells. The hope is that these long-lasting changes will have long-lasting beneficial health effects. But the flip side is that these long-lasting changes might also have the potential for delayed detrimental health effects, such as the development of cancer, as Dan mentioned, has been found in some cases, immunological implications, including delayed immunological implications or toxicities. And that's why long-term follow-up studies are so important in this context to understand the overall benefit risk profile of certain types, many types of gene therapy products. And I'll speak more about that in a moment. There are also immunological issues with some gene therapy products. And again, Dan touched on this. Some people may have antibodies to components, and this may preclude them from participating in a trial or receiving a certain product. Also, once they receive one dose of a cell or gene therapy product, they may develop antibodies that preclude them from getting another dose of the same or a similar product. As we've mentioned, there are also complex manufacturing supply chains and delivery for these products, and administration may involve invasive procedures and significant healthcare provider expertise and support may be required. Also, gene therapy products participants themselves, such as healthcare providers, and in some cases, an institutional biosafety committee might need to be involved, in other words, to protect people, not just the participants, but people who are delivering these therapeutics as providers, researchers, and even family members, for example. And Dan is an expert at that. So if you have any questions, we can go into that in the Q&A. I think another issue is, because of the specialized expertise, people might also need to travel to specialized academic centers. So all of these issues, again, combined, can really, affect the ethics. The other thing is that these products are also developed for in part because of the uncertainties with the new therapeutic class, but also because of the specific mechanism of action. In other words, they're gene therapies, and so they may address genetic diseases or disorders. This class of products is often developed for vulnerable patients with significant unmet medical needs, and for patients often have severe and or fatal disease with limited or no therapeutic options, and in some cases, a short therapeutic window. And again, we're talking in this seminar about the pediatric context, which can be challenging in and of itself. And so, as I just mentioned, there's often a high burden on cell and gene therapy research participants, their families and caregivers, and this has to do with the specialized expertise, the manufacturing and supply chain issues, often meaning that there might be limited geographic availability for sites. And again, we've also really mentioned this long-term follow-up issue as well, which I'm going to go into in another slide. But really what this boils down to is that there are truly profound implications for deciding whether and when to participate in a gene therapy trial and even receiving a gene therapy once it's approved. There may be opportunity costs. You may not be able to receive another gene therapy in a research or clinical care setting. And because of the durability of the product, you can't really necessarily, it's not like taking just a regular medicine or a pill where, you can just stop taking it. You may not be able to withdraw in the traditional sense because of these long-lasting effects. But on the flip side, there may also be eligibility restrictions to trials like inclusion and exclusion criteria, and there might be a sense of scarcity and a limited number of spots. And the opportunity for changing the trajectory of some of these serious diseases may be limited. And so there can be a real pull for patients and families to participate. So all in all, it can be a really complex and emotional decision on whether to participate in a gene therapy trial or even to receive an approved gene therapy. And I know Merritt's going to be talking about her personal experience in just a minute or so. So the other thing I just want to point out is we kept talking about long-term follow-up studies. And so I think, just to offer a little bit more perspective on this, there's widespread agreement that long-term follow-up studies are important and necessary in the context of cell and gene therapies because we have uncertainties about long-term adverse events, and people really want to understand when they decide whether or not to participate or receive a gene therapy. They want to understand what are the overall risks and benefits, right? Even the long-term ones. But doing these studies can pose scientific, financial, operational, social, and ethical challenges. That even though this is not a new problem, actually, this, long-term follow-ups and the challenges have been, in the literature for a long time, and we've known about it, but it's becoming even more of an issue, as Dan showed, as we do more and more cell and gene therapy research and more products come to the market. And one of the specific challenges is figuring out how much data to collect. So that sweet spot of, it's really focused on safety, but then families might want more data about efficacy. That's also important, but it's like the burdens and the trade-offs are hard to judge and need to be collectively figured out. And also the data that comes from long-term follow-up, in order to maximize its value, really should be shared and not siloed. So what can we do about all of this? It's easy for ethicists to point out the challenges, and it's a little harder to point out the solutions, but I think it's clear that cell and gene therapy research requires thoughtful consideration about equitable access and appropriate trial design. And in this, and this is because there's this high stakes patient context in combination with the considerable scientific and technical complexity of the products. And so what does that mean? So it means, when researchers and ethics committees think about the protocols, there needs to be robust justification of eligibility criteria. There needs to be a focus on, thinking carefully about where the trials will be located and, if possible, enabling geographic access. It means also very careful consideration of the starting dose and the escalation plan. If this can only be given one time, then each opportunity, from a research basis, we're just trying to find out what works best, but from each particular participant's basis, it's their one shot on the goal. we need to think carefully about what outcomes are going to be measured. You know, there's the different approval pathways with surrogate markers, but what outcomes are meaningful to the families? Also, the use of controls, whether controls are used or not, is very significant for families, of course, with these limited therapeutic windows. And of course, you know, all these decisions are very complicated and different perspectives come into play with different parties have different incentives within being in the research. But of course, we're all trying to aim for the same goal, which is to get therapies that work to patients. So okay. And then also we can also appreciate that a robust informed consent process is absolutely critical in this context, but also that it can be extremely challenging to communicate the complexities of the science. Dan did a great job. What is involved, alternatives to participation, and the nuances of the known and unknown risks and benefits. And it's really important to ensure that decision makers understand and have realistic expectations. So differentiating between optimism and hope, which are appropriate, from misunderstandings and misconceptions. So in this context, informed consent, you know, should take place over a series of conversations, allowing time for reflection and questions. And then last but not least, I just want to point out that ethical design and conduct of cell and gene therapy research requires respecting and involving patient communities in trial design. Their input is so important. And as we've said, we need to make efforts to minimize burdens on research participants and their families as much as possible, and also maximize the benefits. So hopefully this overview has provided some food for thought on ethical issues that arise in this context of research on cell and gene therapies. And, you know, I think the ethical development of these products is truly a collaborative effort and a shared responsibility among sponsors, researchers, review committees, clinicians, patients, their families, and many others. And thank you so much.
Thanks, Caroline, for sharing your expertise in this field. It is truly invaluable, and we appreciate you taking the time to join us today. Now we are going to circle back to Merritt. Let me just go to her slide. And I would like to begin by letting you meet Brecken. He was diagnosed with Duchenne's muscular dystrophy just shy of his 3rd birthday. At that time, the life expectancy for DMD was in the late 20s, and many of these children may never walk or even sit up. And at the time, there was no cure. After Brecken's parents received this diagnosis, they began a diligent search for treatment and care. Here today is Merritt, Brecken's mom, to share his story and the profound impact cell and gene therapy had on their lives. I'll turn it over to you, Merritt.
Great. Thank you so much, Angie, and to IAC for the opportunity to share my family's story about raising a child with a rare disease who received gene therapy. Our firstborn child, Brecken, was diagnosed with Duchenne muscular dystrophy, which is a rare genetic condition just shy of his 3rd birthday after a very long diagnostic journey. And our concerns with Brecken's gross motor development began when he was delayed with crawling and walking. And when he did walk, he tripped and fell often. He always struggled to really lift himself off the ground and walk up the stairs. And it was difficult for him to keep up with other kids his age on the playground. And we noticed that he walked differently from the rest of them. So we pushed for answers, answers from our pediatrician and other medical professionals, but our concerns were dismissed for nearly 2 years. And I can tell you as a mother, it's so deeply distressing to experience being told that nothing is wrong with your child when you certainly know that something is. It wasn't until we took Brecken for a second opinion at a children's specialty center here in the Twin Cities that we received his Duchenne diagnosis from a neuromuscular physician that really changed our lives. Duchenne is the most common gene-linked disorder in boys. It occurs in about 1 in 5,000 male births, and it's caused by mutation on the DMD gene that codes for dystrophin, which is a protein that's critical to muscle function and preservation. Those affected with Duchenne often lose their ability to walk by around age 10 to 12. They also lose the ability to feed themselves and to breathe independently. Most succumb to heart or pulmonary failure in their early 20s. When our neuromuscular physician delivered the Duchenne diagnosis to us, he told us that there was reason to be hopeful. Now, at that time, early stage clinical trials were just starting to test gene therapy in boys with Duchenne. For a child living with a progressive muscle wasting disease, time is muscle. Every single day matters to our boys. So for our family, the choice was clear. We knew with certainty what would happen if we did nothing to intervene. So for the next several years, we contacted gene therapy clinical trial sites to add Brecken's name to the interested participant list to try and get him in. Then, in the spring of 2019, after screening for one of these trials, we received a phone call from Nationwide Children's telling us that Brecken was invited to participate in a phase 2 double-blind placebo-controlled gene therapy trial, enrolling only 40 boys. So when we packed our bags to drive to Columbus, Ohio, I was 6 months pregnant at the time with our third son, and we had to leave our 2-year-old son at home, which was very difficult to do as a family. But our hope was that this opportunity could possibly change the course of this devastating disease, not only for our son, but for all children living with Duchenne. During the informed consent, we understood that there was nothing guaranteed, right? The trial may fail, or there could be significant side effects or adverse events, including death. We knew that even if this trial succeeded, that the durability of gene therapy and the extent of the functional benefit were unknown. We also knew that this was likely to be Brecken's only opportunity to receive gene therapy in his lifetime because of the antibodies that he developed to the virus used to transport the gene therapy into his body. Knowing this, my husband and I still chose to sign that informed consent because this clinical trial represented hope for a different future for our son. So in May of 2019, at 5 years old, Brecken was admitted to the ICU at Nationwide Children's. He was surrounded by his dinosaurs and coloring books and stuffies. And when the team was in place and the infusion began, I remember sitting next to Brecken and putting my hand on his leg as he played with all his dinosaurs, like listing off their scientific names, many of which I still can't pronounce to this day. But I remember also thinking, like, he was too little to fully understand how monumental this moment was for him and for science and the entire Duchenne community. Brecken received placebo that day, but he was dosed with gene therapy a year later in the spring of 2020 at 6 years old. And after gene therapy, we saw a dramatic improvement with his stamina, his ability to get up off the floor and climb the stairs and keep up with the other kiddos. And his North Star ambulatory assessment, which tests the functional motor abilities of children with Duchenne, increased. And the gene therapy that Brecken received in that clinical trial has now been broadly approved by the FDA for children living with Duchenne. So today, Brecken is nearly 11 years old and he's in 5th grade. He's traded his dinosaurs for football and is a diehard Buckeye fan, a love that he shares with the neuromuscular physician at Nationwide after nearly 6 years now, I think, of us traveling to Ohio for the clinical trial and now the extension. We are grateful that Brecken still gets through a full day of school without any accommodation. He loves playing football and soccer in the backyard with his 2 younger brothers and the neighborhood kids. And he was the assistant coach on the 5th grade football team this year, helping to run practices and call plays. While the stairs are starting to become more difficult for him now, he's now enrolled in another clinical trial for a possible add-on treatment to gene therapy, with the hope that it will further stabilize his disease. His trajectory does look very different than most boys living with Duchenne at his age. We are grateful every single day for the opportunity that we've been given to participate in clinical trials, but there's still so much work to be done. Many individuals have pre-existing antibodies to the virus for gene therapy and can't receive it, or are otherwise excluded due to their genetic mutation. And others just haven't been as responsive as Brecken has been to the therapy, so we cannot leave these families behind. With Brecken, my husband and I have always emphasized the importance of advocacy and in the power of him being able to share his own story to affect change for all children living with Duchenne and all individuals living with Duchenne. This leads us to Capitol Hill and the Minnesota legislature each year to advance national and grassroots advocacy initiatives that really positively impact the rare disease community. Our advocacy has also focused on patient access issues, newborn screening, drug approvals, and patient-centered clinical trial development. Today, I can tell you that I am more hopeful than ever that we are entering this new era for individuals living with Duchenne, with 8 approved therapies, including a broad label for gene therapy. But we know that not any one therapy is going to end this devastating disease, and that we're going to need to take a multifaceted approach utilizing a combination of therapies to treat this disease. Next year, we're going to be launching a groundbreaking collaboration called the Dystrophinopathy Clinical Research Network that's going to leverage existing Duchenne clinical trial networks and infrastructure and expertise across the country to really revolutionize how we develop therapies to combine them to slow the progression and loss of function for our children. I can keep the faith and hold on to hope that Brecken's future will be different because of the remarkable individuals that we're supported by, the clinicians, the scientists, the researchers, hospital staff, and industry members who get up every day and go to work to fight for our children. Please know that we are so grateful for you and all that you do and the vital role that you play in our children's lives. Thank you.
Wow. Thank you, Merritt, for sharing Brecken's inspiring story. Your journey is a testament to the resilience and hope of these innovative therapies. I'm truly touched by your family's courage and determination. And as you mentioned, I also wanted to take a moment to acknowledge the dedication and hard work of all professional and stakeholders in the field of gene cell therapy. Their commitments and efforts are crucial in supporting and advocating for all these treatments. And it's always important to remember as we continue to work in cell and gene therapy that we remember behind the science, there are children that can't wait. So for now, I think what I would like to do is stop sharing. We have quite a few questions that I think we'll try to work our way through in the time that we have. The first question, and I think all 3 of you mentioned it, is this long-term follow-up. Specifically, how is that realistic? How are scientists expected to follow these families? What are the expectations on the part of the family being followed? There's a lot of different caveats, the ethical concerns about when child turns 18, do they have a responsibility to be followed? So I think what I'll do perhaps is start with Carolyn to kind of give that overview again about what she mentioned, and then maybe get Dan's perspective, and then Merritt, certainly we want to hear from the family about what this looks like.
So thank you so much, Angie. I think long-term follow-up, at the MRCT Center, thinking about issues, one of the jobs that we have is identifying challenges, and that has definitely risen to the top of one of the, challenges that needs collaborative effort. And the reason for that is that, as I said, many different stakeholders want to know the long-term follow-up outcomes in terms of safety and efficacy. Of course, patients and their families, also payers, regulators to have their to fulfill their responsibilities of bringing safe products to market, or approving safe products and ensuring their safety. And also investors. So it's a big issue with a lot of different people agreeing that they want the data, but then it's challenging to actually do it. And so there's long-term follow-up in the context of clinical research participants. So when a drug is pre-approval, there's a need to follow people along. There's also even sometimes a need to follow up people who have received approved product because the trials might be small in number, and also potentially the clinical care setting might be different in certain ways from the actual research setting. So there's a need for that follow-up in that setting as well too. So for companies, and particularly small companies, this can be challenging because it takes resources and money and coordination, and it can also be extremely challenging, perhaps even more challenging for academic investigators who are just trying to sort of do the initial research and maybe potentially first in human trials for very innovative gene therapies on a smaller scale. How do they, if they're just, an academic investor trying to push things forward, actually do long-term follow-up studies that are 5 years, 15 years, or potentially up to a lifetime long, depending on the risk profile of the product. So just logistically, it's very challenging. And then, again, I mentioned this in the talk, but it's like, how much data should be collected, coordination and data sharing. Those are some of the, the things. And I think what we're seeing is that to do this well, potentially, there might need to be, thought about a new system. This is like thinking pie in the sky, but for doing long-term follow-up and sharing data, and figuring out what outcomes across, different products or diseases should be followed so that we can compare across, single long-term follow-up studies as well as just within them.
Sure, I completely agree with Carolyn. She brought up some excellent points. Not only is it logistically difficult, particularly in the case of lifelong, long-term follow-up, there's a considerable cost associated with that. And let's say, for example, let's say we have a biotech company, so we're not even talking a large pharma sponsor, that they're developing their product, they're doing phase 1 trial for their product, and at some point after the trial starts, the biotech company goes out of business. Who would then be responsible for the long-term follow-up of those subjects that require potentially lifelong follow-up? So there's a lot of caveats and details that need to be worked out.
That is a really good point. I just really hadn't thought about that particular scenario. So I think that there are a lot of things to think about. So Merritt, now I'd like to hear from the parent's perspective. As being a clinical research coordinator many hats ago, I honestly didn't know how to do this with families. It wasn't clear. Pharma protocols didn't kind of set the expectation about when, where, how you would share follow-up data with parents. So I'd love to hear your experience so far.
Yes, thank you, Angie. Well, I think, data sharing is a big issue for families, right? Especially with these extension studies where in a gene therapy, you're not on active therapy. We don't have access to our data. So it puts families in a situation where they need to repeat a lot of testing from their clinical trial visit, also then within the clinic. So what I would love to see is that there are robust data sharing policies that these companies put in place, and that patients are able to access the PT assessments, the blood work, the other the cardiac assessments that are done, cardiac MRIs, that we're able to actually take those and use those in the clinical setting, so our kiddos aren't having to repeat those tests. It's a significant burden on families to have to do that, and it's not really outlined by the companies how we get access to our own data.
Yeah, and one of the questions that's being asked is, who's thinking about these details? I don't have a good answer for that. They want answers to Dan's question, and I think these are things that are still being considered by regulatory agencies There will be requirements, I'm sure, policies put in place for pharma companies conducting cell and gene therapies, but I don't think we have an answer. And I think that's part of the concern right now, is that there's not a streamlined way of handling these different scenarios that could arise. And, that can be very scary, especially if you are in the industry and it's not comfortable not to have an answer, right? It certainly is an example of the technology is ahead of the process almost. But as was mentioned, it's life-threatening, so a lot of times we proceed and kind of have to work our way backwards, unfortunately. So one of the other questions that was asked was about real-world data. So it was about the use of non-interventional studies to kind of collect real-world data, and how what are the ethical concerns about collecting the data in that fashion? I'm not sure I'm doing the question justice because it is now gone off my screen. Ethical issues concerning using non-interventional data. Carolyn, do you have anything that you might be able to add to this?
I mean, I'm not exactly sure what the question is getting at exactly, but I think that long-term follow-up is not interventional in the sense of, the recipient, the patient have already received the gene therapy, and so it's not that. But at the same token, sometimes they can involve invasive procedures to test different aspects. For example, Dan said, looking at the cells, whether the vector has, whether the DNA has integrated and where, or other aspects that can, need potentially invasive procedures. That being said, I think if you're just talking about the data that's generated, I think Merritt's comments about, collection of data, sharing of data, ability to do meta-analysis, while also protecting patient privacy, is challenging. And I think also, the interim results, both from on an individual level, as Merritt was saying, and also an aggregate for the patient communities, are challenges that I still think need to be solved in long-term follow-up, and perhaps, not to say, not for the pun, but broad consent for secondary use of data in general and all types of studies probably also needs, solutions, and actively being talked about there too. I will say to Dan's question about, what happens if a business goes out of what happens if a biotech goes out of business? In a recent webinar that was co-hosted by the MRCT Center and PGTME, we on long-term follow-up, and a question came up similarly, and there is guidance that says that, businesses in that case should consult with the FDA, and the FDA representative said they do try to work with the investigators associated with the trial to sort of get the long-term follow-up studies to continue, because otherwise, if they're dropped, then obviously the opportunity to understand the long-term benefit and safety particularly is compromised.
Wonderful. So there's been some other questions posed. Unfortunately, we have run out of time for questions. I know that all three speakers are open and willing to address any questions that you may want to direct to them. I think that the biggest question that was posed at the end was about access. And honestly, that could probably be its own webinar in terms of having enough, how our insurance company is going to pay for it. How are we going to scale it? Not every hospital is doing cell and gene therapy. It takes a lot of resources and training. So maybe that will be an idea for a future webinar. So I really want to appreciate our speakers today and the time and energy that they have given us. It's invaluable. And we just have a few housekeeping things as we wrap up. And I can see. So in closing, I do want to remind everyone that this field is constantly evolving, and there are new developments and discoveries being made at a pretty rapid pace. So it's important that we all stay educated and continue to attend webinars like this so we can stay up to date and have the information and education that we need to provide to our families. So we want to thank you for attending. As you log off today, you will be given a link for a brief feedback on the webinar. We really appreciate that. We want to continue to make sure we're providing relevant content, and we appreciate any feedback that you have about the webinar. Speaking of webinars, we have 2 future 2025 webinars that will be presented in March and June respectively. We will be taking a look at pediatric diversity. That will probably address some of the access questions across the board regarding pediatric clinical trials. And then we also will be presenting a webinar on policies and regulations surrounding pediatric research. If you would like to visit our new website, we would love to have you come see it. We feel that the changes that have been made make our mission clearer. And you can also find all the recordings of previous webinars in our learning section. If you would like to request a certificate of attendance, you may do that today by reaching out to us. We would ask that you try to do this in the next 30 days just to help us keep organized. And that a reminder that this is a certificate of attendance that you will submit to your institutions or regulatory agencies to request continuing education credit. We are not a CE provider, but many institutions will give you 1 hour of CE credit for participating today. We would like to thank Gilead Science. They are supportive of our educational webinars. We appreciate their commitment to pediatric research education. And lastly, thank you everyone for attending. Thank you again to my speakers. We look forward to offering you future educational opportunities. Thank you, everyone.
Thank you.
Thank you.